We're going to proceed with our next speaker. I'd like to introduce Adam El-Hafi, Chief Science Officer of Essential Pharmaceuticals, LLC. Adam is going to talk about an innovative approach to decreasing costs, increasing yield while improving quality. Please welcome Adam.
How's it going? So essentially taking a closer dive in at what these gentlemen just discussed about manufacturing. So this is a novel tool basically to increase productivity and to also have good protein quality. So these gentlemen were just talking about manufacturing, but when in reality we're talking about cells. And so when these cells start to get cranked up, they start to have deficiencies, and those deficiencies end up in having different protein quality, different aggregates, loss of consistency and quality of protein. And so it's been a conundrum that try to figure out how do you improve the quality of both of these things. And so we have a novel technology that kind of addresses that. And so I'll go through that. I'll warn you, there's data slides in here, but I'm gonna fly through those, but more so just to prove the point of what we're trying to discuss. So first of all, who is Essential, this is what we're gonna discuss today. Who is Essential Pharmaceuticals? How lipids play a role in this topic that I just discussed, in increasing productivity and increasing product quality attributes, a look at yield and productivity, glycosylation, and then a discussion at the end. So Essential Pharmaceuticals, we've been around for over a decade. Our primary product is actually in the transplant space, so we come at this manufacturing from a 510K approved drug vision. So everything about the cell S and the lipid component all has all of the attributes that you would expect from a drug. So it's made GMP, it's animal component free, and it's quality controlled, not like a lot of the other media necessarily go through. But we take that extra step just because that's the backbone of our company, and we've been around for quite some time and quite good at doing that. So Cell S, as you see, it's chemically defined, and we also have another product called Clen S. So here's basically the meat of what Cell S tries to accomplish. As I said, lipids is the novel way of delivering lipids is what Cell S does, but all of the protein bits of manufacturing all happen on lipid membranes. So the ER, endoplasmic reticulum, is where proteins are synthesized, and then they're shuttled out to the Golgi in a membrane vesicle, and then post-translational modification, so this is where your protein quality attributes happen here, and in each section, different glycans are added, and so sialation and galactosylation happen in this portion, and then after that it's secreted. What happens is that when you go to push increased titer, and you go for more cell divisions or you go for higher cell density, the back half of the Golgi stops working properly. And so what that means is that you don't get your proper glycosylation, and you don't get proper protein folding, you get more aggregates, and so then the secretion also doesn't necessarily happen as well. And so one of the biggest things that people are talking about now is sialation. It's been discovered the more sialated a product is, the longer that it will stay in the blood and the more you have active monoclonal antibodies. And so this is where Cell S comes in, is that conceptually people understood that this is important, that you should address the addition of lipids and addition of lipids in the right ratio, but they've had a difficult time doing so. And so essentially the issues with adding lipids is that they are not soluble in media. And so when people have tried to do it, they've had precipitation. Precipitation have led to clogged filters. And then the other half of it is that then you get oxidation. And so oxidized lipids inside of a cell culture vessel will start to destroy the cell. And so there's a lot of stories where people try to add different either free fatty acids or cholesterol, and anything that lasts more than 3 days, you start getting oxidized free fatty acids, which start to kill all of the cells. And so then nothing works. You get no protein and your cells are all dead. And so that's a problem. The other issue is that you start to get degradation of those free fatty acids, and then they adhere to the vessel wall. So this adherence to the vessel wall was shown by a group from Merck, and they showed here, and so again, one of the topics that they were talking about is using these 2,000 liter bags, the single-use bags, and so the films on those bags are either EVA or PE. And so what the Merck group tried to understand is, can you add lipids into either one of those bags, so if you want to do any of the single-use cultures, and they found that after 72 hours, the cholesterol is pretty much 90% gone in the ways that people traditionally add cholesterol in, which is either ethanol bound or cyclodextrin, or ethanol solubilized or bound to cyclodextrin. And then the free fatty acids of linoleic acid and linolenic acid, they're bound up by the film in 6 hours. So addition of lipids hasn't been possible in any of the single-use bags. And so even further, they looked in the traditional vessel, so they looked in steel and glass, and after 72 hours, the cholesterol is pretty much gone. It's 75% gone. And so for 14 days, which is a traditional batch culture, essentially your lipids are gone, and in anything that's going for 200 days, which is the continuous bioprocessing, you can imagine the lipids are either gone or oxidized and causing complete destruction. So the addition of lipids has been a difficult issue to do, and that's what CellS overcomes, and that's the new technology that CellS provides. So, as I just said, one of the benefits, though, is that you do get consistency because the cell starts to work better. And so we have the patent that covers this unique lipid delivery technology, and the way that it's designed is that you don't have to change any of the current processes that are going on. So whatever media backbone or whatever feed structures you're currently using don't have to be changed, and that was done on purpose. That way, this can be brought in at phase 1 or phase 2, and people are already using it in preclinical work right now in their process development. But it could be brought in later because it already cleared the GMP animal component free and doesn't change any of your process parameters. So we'll take a look at yield and productivity to show that it works. And so one of the first things to think about is what are the ways that people try to increase yield now. So you either use media additions, more cells, so this is a perfusion, so you double titer what people typically have been doing as they quadruple the cell density. So 4 times as many cells gives you twice the titer. Problem is then you have more host cell protein, and you also have more cell debris, or people optimize the cell culture process. One of the things people never tried to do is actually improve cell functionality. So if you improve the lipids, the ER, the Golgi, the lipid vesicles that pass through, that approach hasn't been taken, and so that's where CellS comes in. And so the way that all of the experiments were set up and the way that we always do things is actually we leave them open-ended, and we always use an outside contract group so that we don't have our fingers on any of the results. And essentially the way that they're done is that you could add CellS, do an optimization scheme, and there could be a pathway where it comes out poorly, an optimization where no matter how much CellS you add, you're going to have a benefit, or there could be a window where it's beneficial. So the first thing that we did was actually see whether or not there's any effect of CellS on any of the process parameters. Well, I'll give you a preview of the data that we have increased titer and increased productivity. And so one of the things that we did was just first show that by adding lipids, you don't affect any of the process parameters. So your viable cell density is the same, glucose the same, glutamine, ammonia, lactate, glutamate. A lot of charts that all say that addition of CellS does nothing to any of your process parameters and nothing to any of the outcomes that people traditionally look at for optimization of increasing titer or by changing, trying to affect their glycan profile. None of those aspects are changed. So essentially making the argument that addition of lipids uses a completely different mechanism than any of the glycolytic pathway mechanisms that people traditionally use to try to increase the amount of titer. And so this basically shows that we do get increased titer, and we do so across different media backbones, so Sigma, LifeTech, Irvine, and Lonza, and these are all increases of yield by 20% or more. And this basically shows that we're able to scale this. So this was done in an amber. We also did it in a wave bag. We've done it in a steel tank of 3 liters. In groups now, we're taking it into 250 liters and scaling this up further. So this increase in yield is repeatable in different clones, different media backbones, and is also scalable and has been used in single-use bags. So the way that we achieve increased yield is basically by increasing productivity. So since VCDD doesn't change, but the amount of protein goes up, and that happens both on a per cell basis and if you look on it by QP. So if you look at the integral under the curve and the amount of protein that's made, by either metric, there's an increase in productivity. So essentially it argues that the cell is working better. And so if the cell is working better, that essentially means that the Golgi itself is working better, and one way to measure that is to look at glycosylation. So how do people affect glycosylation? They look at this and then they say, how do we bring it in? And so you've got nucleotides that you can add in, so you can add in the sugars, or you can modify these enzymes. I'm not going to go into it, but basically they kind of say, well, we can fix the enzymes, we can add in the nucleotides, or we can do some kind of genetic change to the cells to try to pick the ones that are doing what we want. And so that's kind of identified here, but just using pictures and words kind of emphasizes what we're trying to show. And again, the similar kind of approach. What are the levers that you can pull when it actually comes to doing something with your manufacturing process? And so the media additions that people typically add are like galactose, and there are other sugar nucleotides that they use, but galactose is one of the more popular ones. Cell culture processes, osmolarity and pH can drive different glycosylation profiles. The higher osmolarity you have, the lower glycan profile, so you get MAN5s and G0. And different pH, you can eliminate different glycan profiles. Genetic engineering is becoming more popular, especially with the CRISPR technology. So I covered some of this already. And so one of the byproducts of fixing the way that the cell actually works is that you can improve consistency. And so we basically tested that. And so again, don't try to look at this quickly. I just wanted to show that if you do this in multiple replicates, you're going to have a lot of bars. So this is the profile of glycans. If you do statistics around that and measure the standard error around the mean, so basically looking in the consistency of your protein attributes, if you add cell-S, you get lower standard error around the mean. So you have more consistency in your protein quality attributes. The other part of it is that you actually get more of the higher order glycoforms, so more galactosylation and more sialation. And so the reason that the sialation and galactosylation are important, and so again, this, if you remember the picture of the lipids I showed you, this is the cis Golgi, medial Golgi, this is the tail end of the Golgi. These yellow circles are the galactose, diamonds are the sialation, so the more galactose you add, the more potential for increased sialation you have. And so I'll skip this here and go straight to this. This is the punchline.